Hello, folks. Good morning. It is Monday, the 1st of March. We're through February, which means that now we're edging in on spring. It's raining, not snowing outside, so that's a positive move. And we're going to be finishing up our evolution unit just as the seasons start to change. Today, what we're going to be talking about is the process of natural selection again, but now we're going to be talking about a very particular form of it. Now, for this year, in normal years, you would need to know 3 different forms, and we're going to see all of those, but I've color-coded 2 of them out because with the challenges that we face this year under the COVID-19 pandemic, it's not going to be, I think, as important to know those fringe versions that sometimes take effect, but are rarely, if ever, questioned on. So I really want us just to focus on the one primary form of natural selection that we're now familiar with. We're also going to get into speciation, how new species come about. So let's do that. Let's get through the things that we got to do. So, of course, we are in Schoology as we have been for 6 months. We're in Unit 5 for evolution. And here I've started up our Week 4 folder from the 1st to the 5th. And it says Speciation and Directional Selection, which is that specialized version that we're going to be talking about. Inside the folder, I have all kinds of things. I have our notes that we're going to do here as a group. I've got a video, the California salamander evolution, which is really pretty neat. This is actually a natural selection game. And basically, when you play the game, what you do is you pick what adaptations you want. You can add as many as you want to, and then you just play through and see how easy, how hard. Questions like that. But I included it because it looked like it could be a little bit of fun for people to practice these concepts. It's basically the poster project that we did 2 weeks ago, but in more of a game form. And then, of course, we have our lab. This is what we're going to work on Monday and Tuesday and the turn-in for it. So let's get into this, this natural selection piece of things. Now, remember, we're out of Darwin. We're away from Darwin's time period. We're away from his theory. We're away from his life. He's been dead for quite a while by the time all of this came about. So now we're looking at the genetic side of things too. And that takes a very important part of our total theory. So evolution used to be change over time. Now it's a little bit more specific because now we can apply that genetics information. So evolution is defined now as a change in a population's genetic makeup over time. It's changing the A's, the T's, the C's, the G's. It's altering the gene pool. And here you see a wood owl, and these 2 pictures are about 50 years apart. You can just see how, in general, they have become more of a brown color. So it's important to note that the most important thing for evolution to have is diversity, differences, variation. So remember, our genetic variation and our mutations are the key roles. Down at the bottom here, these are all the different colors of giraffe that we have some kind of record for. Now, obviously today, well, we don't have all of these variations still around now. But in having them at some point, that gave giraffe a very broad range of possibly successful phenotypes. So as the environment changes, we want variation because it makes it more likely that at least some group of the species will survive. Case in point, let's imagine that COVID-19 was a much more dangerous virus than it actually is. Like right now, let's imagine that its kill rate was up around 50 to 60% of people who got it. So half of the people who got it are going to die. Like that would be unbelievably bad. That would be like smallpox, bubonic plague level bad. But we've already learned that type O blood people have an advantage against COVID. They do not get the negative symptoms as badly as some of the other folks do. Now, of course, there's more to it. Blood type alone is not a deciding factor, but it does offer an advantage. So were COVID-19 much more dangerous, then we could expect to see more type O blood humans surviving the infection and then passing on that type O blood to their offspring because they were more successful, right? That's a variation that would keep the species moving forward. It may not help every individual, but that variation would let the Homo sapiens survive this environmental change. All right, and there go the lights. So let's look at the classic example, arguably the best example, the peppered moths. You probably remember these. I've already explained it once or twice, but just a quick review. All right, so the light phenotype that you see here was really good because it blended in so well with those white, the white tree bark. Meanwhile, the dark color wasn't. But when the Industrial Revolution hit, all that thick coal soot from machinery and industry floated through the forest, which was upwind and uphill of the factories, and it coated the tree bark. Now, all of a sudden, the light phenotype was the bad one. The dark phenotype was the good one. And so we saw the original population, which was mostly light moths, shift to the dark phenotype afterwards, right? So it moved from here to here because this one, in this situation, was more fit. This is directional selection. This is the most common form of natural selection. This is the one that you have always thought of every time that you have thought of anything regarding evolution. It's always been directional. It has been a situation where when the environment changes, one phenotype, one set of traits has an advantage where the original phenotype can't keep up. And so the whole population shifts. Their gene pool shifts over the course of a couple generations. The ones that don't fit the original population die off. The ones that are more fit, the new phenotype population succeeds, reproduces, passes on the traits that make them successful, and everything shifts over. Now, we could see this in hundreds of thousands of different places, but the peppered moths are by far the best example. Where else do we see it? Well, take a look at this graph kind of behind me. All right, we got two groups. We have predators and prey. We're looking at the speed. And as time passes, remember, this is a lot of time. This is like millions of years time, right? They both started off fast, but not as fast as they could be. Now, the prey were constantly evolving speed because speed makes them better at getting away from their predator. But as the prey got faster, the predator had to get faster. And so these two are locked in this evolutionary battle where they are both going through directional selection just to get faster, to get faster, to get faster. And you can see there are physical changes that appear at those time periods as well. Their legs are getting longer, being the predominant one because longer legs help you run faster. This is directional selection. And this is exactly how most things in nature end up working, especially if the species has a predator. Because if the predator can get you, well, then it's going to constantly push natural selection to select only the best and most fit individuals against that predator. And remember, predators are different. In the moth lab, we assumed that the predator could see colors. We assumed that color was the only sense, that they didn't have any form of smell, that they weren't based on movement. We assumed that the moths would randomly land on whatever was there without any regard for blending in. So just remember, there's a lot more that goes into it than just one thing. But this is directional selection. Now, this is the second form that I'm not asking you folks to know this year, but it's not bad to at least see it. And this is called stabilizing selection. This is another form of natural selection, but now what happens is the low end and the high end of the original phenotype are both not good. The only thing that's really good is the most common phenotype. And so it stabilizes. It cuts away those edges and condenses everything into a much more focused, larger group. One of the examples that they really like to go with is human birth weight. If the baby's too small, it's not formed, there's a problem. If the baby's too big, it can't be born, there's a problem. The next one is disruptive selection. Now, this was always the weirdest, the oddest, the least common to happen. And what you see happen is that old population here is not good anymore. But now both of the extremes, over here and over here, both have an advantage. So you get a split. And that original one group starts to split off into two separate, smaller groups. A good example of that are going to be the sticklebacks. So the stickleback fish used to have kind of a medium-sized, what we call gill rakers. And it means these little prongy things inside their gill so that as water passes over it for breathing, it also catches little bits of stuff for eating, for swallowing. Well, what ended up happening was their primary food source, which was like small water bugs, started to die off. Now, at the same point, you had the larger food sources, the bloodworms, and the smaller food sources, the plankton. And so what happened was disruptive selection eliminated the middle, the medium gill rakers, and instead both the short ones that were better at catching bloodworms and the long ones that are better at catching plankton both now had the new advantage. And so the population went from one group that mostly ate water bugs with medium gill rakers into two new groups, one with small rakers for bloodworms and one with large rakers for plankton. Now, both of these two, stabilizing and disruptive, are likely not going to be important. I will not be testing you on them. They are not generally what the Regents asks you either. And if they do, it's going to be 1 point. So we're going to focus on the directional selection because that's what they always ask. Now, these variations, these differences, this push towards a new form due to natural selection can have some really fantastically interesting effects. So what you see here is the California salamander. Each area is color-coded for what version you find in that area. But what is most interesting is that they are all the same species. These are all the same thing. They can all breed with each other. They're the same salamander. But in different regions of California, we see different phenotypes becoming more dominant, becoming more common in the population. Such as, oh, I actually didn't need to do this. If you guys take a look in Schoology, right here, this first video link is the California salamander evolution. It is a video based on what I just showed you. Really, really kind of cool stuff. And they will show you the salamanders. And this professor that works with it will explain where they're found and why it's happening and all of that. So let's move on to speciation. This is going to be a longer video. All right, so remember, and I know that this didn't copy well, but as this has evolved, they reach these branches, right? One group is well or has high fitness for its environment, so it survives and moves on. Those that don't are eliminated by natural selection. So that means that when we look at something like this, we have to understand that species A is going to evolve into species B, because eventually enough of these changes will build up that it's not the same thing anymore. It's really dark. Hold on. All right. And so A turns to B, B turns to C turns to D. You get the idea, but how? How then do we end up with a situation like this? Polar bears, brown bears, black bears, spectacled, sun, sloth, and Asiatic black bears. These are all currently existing. If 1 turns into 2, turns into 3, turns into 4, then how did we get 1 turning into 7? So this is where we have to talk about speciation. Speciation is the creation of a new species. And generally speaking, that's going to happen one of these two ways. We have our original population, and then there has to be some form of isolation. That is key. Isolation must happen. Maybe it's a barrier, literally like a river or a mountain or something that cuts the population. Or maybe it's something in the population that separates them. We'll take a look at examples of that in a moment. But either way, once isolation has happened, then you're going to start having differences build up. So when we took a look at the Galapagos Islands and Darwin landed there, and he took a look at those finches that so caught his attention, he came to this idea that this one ancestor finch probably ended up there. And then from that, they had changed because they were isolated from one another, maybe on different islands, maybe by their food source, maybe by something else. But whatever it was, it caused changes to build up between each species. The key to all forms of speciation is isolation, right? We've been pretty familiar with this since COVID-19 happened. Stay at home, isolate yourselves, stay away from others. Now, we're going to be looking at 3 forms of it, and I'm going to try to go pretty quickly, so this isn't a 45-minute video. But there's geographic, behavioral, and temporal. And I kind of included a picture for each one. Geographic is the land, behavioral is how you act, temporal has something to do with time. So this is just general, right? Let's imagine we have one group of squirrels. You see them right here, the gray squirrel, right? You have their genetic code underneath. They're one population. That genetic code is shared. It's the same, right? But now let's imagine that a flood tears that forest in half. You have one group on this side, one group on this side, and squirrels are not good swimmers. They just won't if they have any other choice. Well, now you got to understand that this river is the new isolation mechanism. It's going to separate those two groups. And now as small mutations happen, they don't mix. So those mutations are unique to each side of the river. Now, at first, a mutation or two may not make a difference. But as time goes on and more mutations start to build up in those populations, it can ultimately create 2 very different organisms. Now, if they reach a point where their DNA is so unlike each other that they cannot successfully breed, we now have 2 new species, red squirrels and black squirrels. Right? So our one species here split into 2 new species. That is speciation. So, all right, what's the first one? Geographic isolation means that something is going to physically split the population. You know, the river, a canyon, chasm. Keep in mind, isolation geographically really depends on the species. This may sound funny to you, but there are birds out there that will not fly through open air. They will fly through a forest and a forest only, and they will not leave that forest cover. They won't. So that means that when we cut a row up and over a hill through a forest and run power lines over it, we've just created an isolation mechanism. If mama bird is on the left side, but her nest was on the right side, and then all the chainsaws happened and it freaked her out and she flew back and away, and she comes back a day later and that divide is there, she won't cross it. Even though her babies are on the other side, she won't cross it. So now that's an isolation mechanism. For earthworms, building a road is going to be an isolation mechanism. They will not cross it. They don't burrow deep enough to get down by it, because if you're not aware, roads actually go quite a ways down. They're not just on the surface. So things like that, it really depends on the species for what will isolate them. But it splits them into two groups, like the example we just saw with the squirrels. And now you can imagine that each side is going to start to build up its own unique mutations. Behavioral isolation is the same idea, but now instead of something physically separating them, now it's all about something that they do. The best possible example is over here, because country music is terrible. Okay, I think it's terrible. But birds are incredibly specific for what they want in a mate. And if you cannot sing that song that that female wants to hear, nothing else matters. It must be the right song, and it must be the right way, or else there will be no breeding here. Temporal isolation is by far the hardest one to understand, but this is breeding schedules. It has to do with time. So here, once again, Amoeba Sisters, doing great. Says, Ted, wake up. It's time to release pollen. And meanwhile, Ted is saying, give me a few more months, because this flower releases pollen early in the season. This one doesn't bloom and release pollen until later in the season. We have other examples. This is long, but basically it is Romeo and Juliet, but alas, they are two different breeding schedules. And so despite the fact that they love each other, they can never truly be together. All right, so now we can take all of this. We can take all the forms of natural selection. We can take all the different isolation mechanisms that lead to speciation, and we can actually start to really understand this. And to do so, let me give you guys a very real world example. All right, we're looking at bacteria. Here they are. There's a population of bacteria, right? Now, when you get sick with a bacterial infection, you take antibiotics. Now, remember, all of the bacteria have variation. They all have differences. So I put little colored dots in them. We have our normal ones with no dot, and then we've got white and orange and red for the different variations. All right, all of the normal bacteria were killed by that first round of antibiotics. But all of the variant bacteria, all of those with the differences, are a little bit resistant. So then you take a stronger round of antibiotics. That eliminated all of the bacteria with a white circle. So now we're just orange and red. The more resistant ones have still survived. Now, this is unfortunately where most of us fail, because at this point, it's reduced the bacteria to a number that we don't feel sick anymore, so we stop taking the antibiotics. And what does that mean? It means that we now have antibiotic-resistant bacteria that are free to spread. and they're resistant to treatment. If you follow your doctor's directions, you finish off your bottle of antibiotics, you go get the refill, and then you finish off that bottle, keeping to your schedule, then what it guarantees is that you will overload any bacterial resistance. Even stronger antibiotics kill off the next one. If you follow it all the way to the end, even these most resistant bacteria will die. But if we don't, as we often do, and we put that little thing of antibiotics back in the cabinet because we're feeling better, we don't need to take any more medicine, we leave the most resistant ones alive. And this is why we have these super bugs that we have now, bacteria that are immune to all antibiotics. And if you get an infection with them, we just got to hope that your body can take care of it, because if it can't, you're going to die. This is a problem that we have created. We created the selective pressure. We created the scenario that allows them to survive and start to thrive. This is our fault. At the beginning, we had 4 different variations, right? Over half of them were normal, and then smaller percentages were red, gray, and orange. But after that natural selection, antibiotic-based, what we find out is we have now changed their gene pool so that all of them are highly resistant. This could be here go the lights again. This could be a brand new species now. Could be so genetically different from the others that it isn't the same thing. So, 2 rounds of the lights turning off. Ladies and gentlemen, that's going to bring us to a close as far as our natural selection speciation is concerned. I'm going to do a follow-up video going over the lab that you guys will be starting today and working on tomorrow. And I hope that everyone has a great day and that this recording is not necessary, because I will see you in class.